So today we're going to kind of finish off our discussion of some aspects of what happens to B cells when they are in the bone marrow undergoing B cell development. And officially, the process that we're talking about today is more usually referred to as the selection part of the process, but it is still the B cell still in the bone marrow. The B cell still learning how to grow up and become a B cell. We have not actually let it out of the bone marrow to do anything yet. Last time, we talked about these stages of B cell development. Specifically, we saw the 3 stages of the pro-B cell, the pre-B cell, and the immature B cell. And we talked about, in all 3 cases, what job that cell is doing and what was on the surface of that cell. So what was on the surface of the pro-B cell? Sebastian? Nothing. So no BCR. Okay? And then what is the pro-B cell doing? Jen? Making heavy chain. So it's doing the V to J recombination of the heavy chain. It does D to J first, and then it does V to DJ. All right, great. What was on the surface of the pre-B cell? Jen? The heavy chain? Anything else? Jamie? Exactly. So it's the heavy chain plus the surrogate light chain. And those 2 things together are known as the pre-B cell receptor, the pre-BCR. So what types of things are being done by the pre-B cell? Aaron? We're going to make a light chain. So we're going to make light chain. There's one other thing that we're going to do in that pre-B cell, really, before we make the light chain. Yeah, Jamie? Test, yep, and so we're also going to test the heavy chain. Awesome. So then, what is on the surface of the immature B cell? Sydney. The heavy and the light chain, otherwise known as the B cell receptor. So what is the immature B cell doing? Jamie? Testing the light chain. And then today we're also going to talk about the fact that this cell is going to test for self-reactivity. And that's kind of the piece we're going to get at today. There's one other piece that I want to talk about, however. So we can also measure these 3 types of cells using flow cytometry. So I'm going to make 3 drawings, 1 for each type of cell. And with these drawings, we are assuming that somehow you've maybe sorted the cells. So in this drawing, I'm going to draw just what pro-B cells would be like. This one pre-B cells, this one immature B cells. If I really took your bone marrow, I'd see all three of them mixed together. But we're going to just sort of draw it as if I had sorted them out. So again, if I was, say, on an exam drawing flow cytometry plots, first thing I would have to do is draw some axes. And then I would have to label my axes, say which things I was looking at. So this is kind of what do I have an antibody against? Which protein's expression am I looking at on the surface of cells? And very frequently, one of the proteins that we will look for on the surface of a B cell is often depicted as mu. The other one that we often look for is kappa. So if I was getting points on an exam, I would get some points just for drawing this. So what are mu and kappa? Let's think about, have you seen mu and kappa before? Rishi? Okay, yes, you have seen them before. Excellent. Where have you seen them before? Yeah? Okay, so you have seen them before there, certainly, but you've also seen them in this class. Addie? Yeah, this is the IgM heavy chain. So you can kind of think of mu as being heavy chain. Kappa is one of our types of light chain. So you can think of kappa as light chain. And often with our cytometry plots, we look at the data in quadrants. At least we did in the past. People don't quadrant gate as often anymore, but that works for us now. So if we had a purified population of pro-B cells, where in this plot would those pro-B cells fall? Sydney? Near zero. Why would they fall near zero, Sydney?
Because they don't have any of the more kappa, they don't have heavy or light.
Right. We've got no BCR. We've got no heavy chain and no light chain on the surface. So here we can say either yes heavy chain or no heavy chain. So they must be no heavy chain, right? So they must be in one of these two boxes. Here's yes light chain, no light chain. So those cells would be found in a population there because they have no heavy chain and no light chain. And that's in fact what you see if you do this plot and look at pro-B cells. All right, now, if we look at pre-B cells, where are our pre-B cells going to fall? Abby?
So they're going to be in the bottom right.
Okay, why are they in the bottom right?
They have the heavy chains, but they don't have the
Exactly. So our pre-B cells would be there. Heavy chain, yes. Light chain, no. And so that's where we would find the pre-B cells on one of these plots. This one's a little bit of a trick, but it's okay. We'll get through it. So where would our immature B cells fall? Krishna?
the top right.
Okay, the top right. Why are they in the top right?
Because they both have heavy and light.
Because they have heavy and light chains. They're heavy chain, yes. Light chain, yes. So that's here. And that's a great answer. If we are being super technical, there are actually two answers on this one. And that is one of the two answers. Any ideas about what the other answer might be? And I get that you might not catch this one. So yeah, Aaron.
Just a guess, but maybe the bottom right because it's still testing light.
Close. So it is bottom right. These are the B cells. That use the heavy chain and the kappa light chain. However, there are two different light chain choices. Some of our B cells could have succeeded, but not use kappa. They could have used the other kind. And so these are the ones that succeeded, but used the heavy chain and the lambda light chain. So does that make sense? Okay. So this is how we would sort of depict this, and this is in fact how people study this process by flow cytometry. This is how they'll actually look at these different cells. There are some other ways you can do it, but this is kind of the general overview way of doing this. So this figure from your textbook is summarizing a lot of that information. Yeah, Aaron?
So it's not a question about the test. Would you tell us which light chain you used, or would we draw both?
The best answer would be to draw both. So we're basically here now talking about what happens to those immature B cells today. So we are really kind of with the immature B cell. We have not yet let it leave the bone marrow. So we're still up here thinking about what exactly that immature B cell does. And one of the things that we talked about last time was the testing of the light chain. And we saw that the B cell got 4 tries to make a functional light chain. So after it makes a heavy chain, whether it's on the first try or the second try, then it tries to make a light chain. It tries kappa first, and it might do kappa correctly or fail. Then it tries the other kappa, might do it correctly or fail. Then it tries lambda or fails. Then it tries lambda again, tries lambda or fails. At this point, it dies because it has no more tries. So that's how you can see the ones that succeeded with kappa and the ones that succeeded with lambda on this plot. So this is kind of where we left that B cell last time. But we've got one other piece of all of this. So when I've told you about clonal selection before, this is the way I've depicted clonal selection. Every part of this that we've seen thus far has had nothing to do with antigen. So we've made a heavy chain. We didn't care what antigen, we just picked a V and picked a D and picked a J. We didn't care if that was for any particular antigen, we just picked them. And then we tested it to see if it made a protein. We didn't care what it bound to. We made a light chain. We just picked a V and picked a J, made a protein. We didn't care what it bound to. So all of this has been going on in the absence of antigen. However, in making that dramatic diversity, in reality, what we see is this. We make this massive diversity. Here's only 4, but it's really like millions. And we make such a diversity. We make basically a diverse enough set of cells that we have one cell that can bind to pretty much every biomolecule. We make one antibody of everything. This is actually super important. If we were going to wait until after we got infected to start making things, we'd never win because the microbes would reproduce so fast. So we basically make one of every possible option to start with. But every possible option includes things that are reactive to self. B cells that make antibodies that can respond to self-antigens that could lead to autoimmunity. So we make them all. And then we go ahead and we test to see which ones bind to self-antigens. And we kill those B cells that are responding to self-antigens, or we get rid of those B cells to make sure that we no longer have self-reactivity. And only then do we release the other cells from the bone marrow. And so this process is kind of the process we're going to be talking about now. This process is often known as the central tolerance, is a process of central tolerance. And so in fact here you can see the overall clonal selection hypothesis. So we have these B cells, Each B cell has a bunch of copies of the identical receptor. That's thanks to allelic exclusion. Yay. On stimulation, the B cell will migrate and it will replicate and make a whole bunch of descendants with that same receptor. So that's this part. Replicate, make lots of descendants that match. At the end, we have more than we started with. And receptors for self-antigens are deleted during development. Here it says embryonic development, but honestly, you're developing and deleting cells today. I actually don't know why the book says embryonic development, because it's really development of the B cell whenever in life it happens. Tolerance is a word that immunologists use to mean ways we get around having immune responses to self. We tolerate self. So it's how you sort of avoid autoimmunity, how you don't make responses to self. There are different kinds of tolerance. Central tolerance, which is what we're seeing an example of here, is a type of tolerance that happens while cells develop. It's the type of tolerance that happens in a primary lymphoid organ. So as B cells develop, they undergo central tolerance processes. Later, when we see T cells developing, they will also undergo central tolerance processes. As we go through this process, you will see that there are some flaws in this plan. There are some places where you could be like, but, that I can think of something that could mess this up. There are some places where central tolerance is not perfect. That's okay, because we have other forms of tolerance that we get to use as backups. They just happen later. Yep, Sydney. Failures in either central or the other type, peripheral, are what cause autoimmune disorders. And in fact, there are specific hallmarks of autoimmune diseases that are central tolerance failure diseases or those that are peripheral tolerance failures. What you will generally see with this process is that I like to kind of think of the fact that the way the immune system is set up, and this is the beginning of us thinking about that, is that we are always kind of balancing on the edge of autoimmunity. We want to have the broadest immune response possible to protect us against every microbe. And so we go right to the edge of being autoimmune. And so you can see kind of all the things that are protecting us from being autoimmune, but the fact that it's a tough line. And central tolerance is kind of the first place where that line happens, but you will see, you're like, oh, but that's an obvious problem place as we go through this. Yeah?
So if you have an antibody for everything, then what's stopping the antibodies against the, like, for example, the beneficial microbe from causing these things?
That is a fabulous question. There are many scientists who would love to know the answer to that question. So what happens with the antibodies with beneficial microbes is Sebastian's question. Why don't we get rid of all those beneficial microbes? Some of that is going to come up in peripheral tolerance mechanisms. And so in some cases, we don't necessarily get rid of the antibodies against them in the bone marrow, but we might turn those B cells off later. And say, no, I don't want those antibodies. Those antibodies are hurtful. So some of it's peripheral tolerance, some of it is unclear. But great question. All right, so we started to understand the central tolerance process with a number of different experiments. And what was discovered in these experiments is that in the case of B cells, I said that very specifically. In the case of B cells, there are three things that can happen if a B cell is autoreactive. There are three mechanisms of central tolerance in B cells, which this was described in these experiments. I've already mentioned one of them. And so one of them is known as deletion. And so deletion just means that the B cell reacts with a self-antigen and is deleted. It undergoes apoptosis. This is actually really sort of something that people have a lot to think about in the field. And one of the reasons why this is really interesting and why there's a lot that we actually still don't know about deletion is because the signaling is a little bit hard to understand of how this works. Because what's really happening here is the B cell is getting a strong signal and it's dying. But if it got the identical signal outside of the bone marrow, it would be like, woohoo, antibody time. So somehow this B cell knows I'm immature, I'm in the bone marrow, and I saw my antigen, so I should die. Versus, I'm mature, I'm out of the bone marrow, I saw my antigen, I should live. And how the heck the B cell knows that difference, we're actually not very sure. Because it's the identical signal the B cell is getting. And so you can see this is happening when that B cell is seeing antigen in the bone marrow, not when we're out in the periphery, out in the rest of the body. So somehow the B cell is getting some other signals in the bone marrow that say, you are in the bone marrow. You should not be seeing anything right now. If you are seeing something right now, it might be collagen. And that is bad, so you are going to die. Notice that the only antigens that should be available for this process are antigens that are in the bone marrow. So an antigen in your eye, for example, is not going to trigger this. That B cell that might make an antibody against a protein in your eye, it can't get caught here. It's going to get to leave because its antigen isn't in the bone marrow. And in fact, all three options that we see today have that same flaw. They only allow for testing against antigens that are present in the bone marrow. So any other antigen that's present elsewhere, so basically anything that's specific to a tissue that's not the bone marrow, we're out of luck. So, things that are in every tissue, like, actin, we get rid of those B cells here. But things that are specific to, individual tissue specifics, we got nothing. And that's where some of our other forms of tolerance are going to be really important. So this was kind of where we were at. And then some experiments were actually done to try to understand the central tolerance mechanisms of B cells even more. And there were a lot of arguments in the field about central tolerance. In fact, there were some T cell central tolerance experiments done at the same time as this. Everybody was having the same argument. So if you were going to do some flow cytometry, let's say you had an antibody against heavy chain. And you had an antibody against the antigen-specific B cell receptor, okay? So let's say I decided to look in my mouse to see, does my mouse make antibodies against SARS-CoV-2? I would find some B cells up there. They have a heavy chain, and they're specific for my SARS-CoV-2 antigen, right? Okay, so what am I going to see if I screwed up the experiment? Sebastian. Maybe. Or maybe I'd see nothing, right? What would I see if we assume deletion is what happens to B cells, and I look for B cells against a self-antigen? Yeah, Jamie. You would hope to see nothing, right? You would hope all those B cells are deleted. And so some people would do experiments, and they would see nothing. And other people would be like, you just did your experiment wrong. It's not really nothing, so you did your experiment wrong. Or they wouldn't have a good way to tell the difference in one B cell versus another. So they would be like, well, my mouse used to have 1,000 B cells, but now it has 999 B cells. And so I think my B cell got deleted. And people are like, yeah, that's not very good evidence. Like, because it's really more like 1,000,000 versus 999,999. And people are like, yeah, I don't believe you that you're actually good enough at detecting that one B cell difference. Nobody could detect that. Even today, you couldn't detect the one B cell difference. Like, none of our machines are good enough to do that. So there was sort of argument about these experiments and what was going on with them. And some scientists came up with a way to try to address that. And so these experiments helped them address those issues. In doing these experiments, the scientists had to make 2 different kinds of genetically engineered mice. They are the 2 mice that are shown at the top. And so I want to tell you about each of those 2 mice. One of those mice is called the transgenic hell mouse. The transgenic hell mouse, so transgenic means that we added a new gene. In the transgenic hell mouse, we added the gene encoding a protein called hell. Shocking, I know. So then what the heck is this? So HEL stands for hen egg lysozyme. The only part of that you actually have any reason to care about is that the H stands for hen, which is a kind of chicken. What this basically means is that we have made mice that have one new protein that they make, and that one protein comes from chickens. So normally this is a foreign protein to the mice, but now they make this chicken protein. That's literally all you have to care about it, is that they make this other random chicken protein. And so in a normal mouse, if I go get a mouse from the mouse store, HEL is a foreign antigen. But in this mouse, HEL is a self-antigen, because it has the HEL protein. Okay? So that's the deal with this mouse. Then there's this mouse, which is the transgenic anti-HEL. In reality, there are actually 2 genes that are added to the transgenic anti-HEL mouse. One of those genes is a pre-rearranged gene encoding the anti-HEL heavy chain. So somebody made in the lab a piece of DNA that has the V, D, and J already put together to encode this heavy chain. And they also made in the lab a piece of DNA that has the V and the J of the light chain, pre-rearranged, and put that in. So these two already rearranged genes are put into the mouse's genome. In this mouse. And so in that mouse, the B cells are going to try to develop through these stages. And when they do that, they might turn on RAG. They might start trying to make a heavy chain to do VDJ recombination. But oh wait, we already have a heavy chain. We don't need to do any more. And they're going to try to make a light chain. Oh wait, we already have a light chain. We don't need to do any more. And so in fact, 100% of the B cells in this mouse are going to respond to this antigen, the anti-HEL. So 100% of the B cells in this mouse are going to be specific for this antigen. 100% of the antibodies in this mice are going to be that antibody. And this is because these pre-rearranged pieces of DNA allelically exclude other rearrangements. So we have allelic exclusion from these ones we've put in, so we don't make any more. You might wonder what happens to this mouse. And the answer is nothing. Because this mouse lives in a lab, which is very clean. We keep all microbes out. So this mouse just runs around doing mouse things. If this mouse escaped, went out into the world, the last time we had a mouse escape our mouse facility and go out into the world, it went to Dr. Dunaway's office and ate his gum. He still talks about it. So if we had the mouse that leaves and goes and eats Dr. Dunaway's gum, it might encounter a microbe, which it would not encounter in our mouse facility. Then it would have no antibodies against that microbe. 100% of its antibodies are against a hen protein. If it encountered hen protein, it would be very well protected. But it might encounter something else. And so if this mouse went into the real world, it would die. But we're not trying to use it in the real world. We're trying to use it in the lab. So it's sort of okay for it. And what we can in fact do is we can do some experiments like this. These are actually sort of the same experiment I was showing you here. If we go and we get a mouse at the mouse store, we can find two populations of cells. So these are actually would be two circles. We find some cells. Here I'm looking basically at the spleen. So I find some cells that have no heavy chain and are not specific for my antigen. Those are like the not B cells. Those might be the T cells or the macrophages or the something else's. We find some cells that have heavy chain on them but do not have the specific light chain. So that's like the B cells against SARS-CoV-2 and the B cells against all the other things. And in reality, we have one cell up there that is a B cell that's specific for this antigen. But when they did this experiment, they didn't have good enough flow cytometers to find that one cell. So you don't see it there. But this is what you would really see in our mouse from the mouse store. And that is what is being shown here. If we look at the mouse that I just showed you, the anti-HEL transgenic, you can see that we have cells in our bottom left quadrant. Those are the not B cells. Those are all the other cells. No heavy chain. And we have a whole bunch of cells up here. We don't have any down here. 100% of the cells that are B cells, 100% of the cells that have a heavy chain are specific for this antigen. And so that's what you can see here. So that's what happens in that mouse. Okay? But this mouse is fine in the lab because it has 100% of its antibodies binding to some random chicken protein. And it is a mouse, not a chicken. So it does not care. And so the key question in this experiment is what happens if you take these two mice and you breed them together to make a progeny mouse. Now, this progeny mouse has this new gene as a self gene, and thus this protein, this antigen, is a self-antigen, hell. So this mouse makes chicken protein. And 100% of its B cells are against that chicken protein. So now it's not like we're trying to find a needle in a haystack of the one B cell. It's 100% of the B cells, right? So if deletion is what happens, what do we expect to see in this experiment, in this mouse? Yep, Sydney. No, okay, so what's it going to look like? Okay, so we'd have basically here. All right, so that's what we might expect to see, right? So they actually, I'm going to show you the data from the paper, and I'm going to show it to you in two different plots. And we're going to predict what we would see with deletion. So in both cases, they do B220. Do it like this. So they do B220, which is on all B cells. And then they either do heavy chain, which is IgM, or they do the ability to bind to antigen. So with deletion, what do we expect to see? No B cells, yes B cells. Yeah. No B cells. So there are only going to be cells that are not B cells, right? And if they're not B cells, they don't have a heavy chain. So that's all we're going to see. Because there will still be T cells and stuff. There just won't be any B cells. What about here? What will we expect to see? Rishi? Would we? So again, we're in the same mouse. Do we see B cells or not? No B cells. So they're still not B cells. They weren't B cells before, and they're still not. And not only do they not have a heavy chain, they do not bind to the antigen. So that's what we would expect with deletion. And this is what we see. So first, these two are those control mice I already told you about. This one is the mouse from the mouse store. Okay? The mouse from the mouse store has not B cells, and this is the one with the antigen, not B cells, and B cells, and maybe like one dot that binds to the antigen, but really, you can't really see anything appreciable, right? So that's what you'd expect from a mouse from the mouse store. And if I look here, we got B cells, or we got non-B cells, we got B cells in the mouse from the mouse store. We don't have any that really, and actually, we won't deal with why they aren't showing up IgM here. Then we can look at the transgenic, the one where we put in the antigen. Yep. It's supposed to represent heavy chain. And yes, there probably should be some here, and we'll not deal with that right now. So let's see. So what do we see in this mouse? This is, again, just the transgenic. We haven't crossed them yet. This is just the one that's making all the antibodies. So what do we see here? Yeah, Sydney. So we see non-B cells. Here we're actually just looking at, do they have heavy chain? And oh my gosh, all the B cells, they have heavy chain. There aren't any B cells down here that don't have heavy chain. Yay, B cells have heavy chains. That's kind of how it should work. What do we see up here? Yeah, Sebastian. Yeah, so the vast majority of our B cells detect HEL, and we'll basically say that's all of our B cells that detect HEL, because this was a little bit of an older paper. So this is papers from 1991. So based like in their level of flow cytometry, that's basically all of them. We'll say, okay? So we'll just say for the purposes of this, everybody detects B cells, or everybody detects HEL, as they should, as I showed you on the previous slide. Okay? All right. So now here's our experiment. This is what we're looking for. And this is what we would predict with deletion. So what do we see? Yeah, Jamie.
We also have effect on a buffer, right?
Okay. So what's this? So these are the non-B cells. So we have non-B cells, as we expected. What's this? Yeah, Sydney. B cells. And what do they have on them? They've got B cells with heavy chains. What? That was not our prediction. There must be something other than deletion going on. We can start to understand a little bit more about what's going on if we look up at these data. So what do we see here? Yeah, Sydney. Yeah, so we've got non-B cells and we've got B cells, but none of our B cells bind HEL. So it's as if the B cells started to develop and they're like, you know what? I think I see self-antigen. I think I want to see a different antigen. Okay, thanks. These B cells have switched what they are responding to. So they can avoid responding to self-antigen. And this experiment was the first time that we had a suggestion that that could happen. And so that led us to understanding the second process, that can happen in order to deal with B cells and central tolerance. This particular one is only possible in B cells. So when we get to T cells later, this is not a choice. So B cells are able to do this thing called receptor editing. And so what you can see is our B cell up at the top. It has little square receptor, little square parts of the antibody, the combining region. And it actually can switch what it has and make something different to become non-self-reactive, to avoid deletion. Specifically, the way that it does that, and in fact, this is a really nice figure from your textbook, is here's our B cell. It responds to self-antigen when it uses the black heavy chain and the black light chain. And so what it is able to do when it does this process of receptor editing is it's able to swap out a light chain. So it can make a new light chain. So you can see here are a couple choices of what it could do. It could make the blue light chain instead, or it could make the purple light chain. Making a new heavy chain is not a choice for reasons I will show you on a future slide. And so here you can see that process of receptor editing in a little bit more detail. So let's imagine that our first B cell, oh, here's my yardstick. Yay. I guess it's actually a meter stick, not a yard, but that's okay. Okay, so we got this B cell. It chooses V3 and J3 in order to make its light chain. So here you can see it does that recombination. So we've got V3 and J3 put together. All this DNA on the outside is still there. The DNA in the middle cut out. If this B cell is self-reactive, it can actually undergo VJ rearrangement again. And it can use a more outside V and a more outside J. So now it can choose number V2 and J4, for example, and just cut out this autoreactive thing in the middle and try another combo. This is only possible because those other pieces of DNA outside were still present. If this cell had started out with V1 and J4 as its first choice, some B cells would do that, then it would be out of luck on this chromosome. It would have used up all its chances on this chromosome. But since this B cell has some choices left, it will, in fact, be able to try to make a new light chain in order to not be self-reactive. And so you can see this process here again. Our cell might be self-reactive. It will then continue to rearrange light chain genes. So instead of using this V and this J, it will delete that and use more outside V's and J's. It will have a new light chain, same heavy chain. But because specificity comes from the combo of the light chain and the heavy chain, we now have a new specificity. And so now this cell will test again. If it's not self-reactive, it's good. It gets to leave. If it is self-reactive, then it might try again to receptor edit. If it's in a case like this where there's still more V's and J's left. On the previous slide, there were not any more V's and J's left. So this B cell couldn't do it anymore on this chromosome. This B cell still has more choices. And so the B cell will actually keep trying to make new light chains to make this work. And in fact, the B cell gets to make a lot of tries because it has 4 chromosomes it can test on. So it can basically try one, try to make a productive allele, try another one. Once it gets a productive allele and a self-reactive, it still can keep trying if there are more and more outside V's and J's, and it can even keep going to other pieces of DNA. So the B cell has a lot of tries to make a light chain before it is sort of completely out of luck and ready to go for deletion. So you can note that this is one place where we are going to do some rearrangement after seeing antigen. So in fact, this is the only reason why this cell might put RAG back on, is just to receptor edit. It is the only time antigen is in any way involved in this recombination process. And once receptor editing is done, RAG is off for the rest of that B cell's life. That B cell is never going to turn RAG on ever again. So one other thing that I mentioned to you is that we only receptor edit at the light chain. We don't receptor edit at the heavy chain. And we need to think for a second about why it is that we only receptor edit at the light chain and that we don't receptor edit at the heavy chain. And so here you can see an image from your textbook that lets us think about receptor editing at the heavy chain. And why receptor editing at the heavy chain does not happen. So take a look at this example of heavy chain rearrangement. These are the steps that the pro B cell is doing. Why is it that once we've gotten past this point, we can't go back and receptor edit heavy chain? Sydney. Exactly. So if you look, the first thing we do is we put together our D and our J, and we get a DJ. And that gets rid of all of the D's that are here between D and J. And then we put V with DJ. And that gets rid of all the D's that are here. The D's are the only ones that have this yellow RSS. So we might have some leftover V's. We might have some leftover J's. There are situations where we could have a V over here and we could have a J over here. That could happen. But if we did, they have matching RSSs. We couldn't put them together because of the 12-23 rule. And the only thing they can go with is a D, and we don't have any more D's left. So in the process of doing VDJ recombination of the heavy chain, we delete all of the D's. And as a result, we cannot receptor edit at the heavy chain locus because we don't have the things we need in our RSSs here. All right, so all of that is this second option, which is the receptor editing option. But we also have a third option, which was also described in these experiments. And in order to understand this option, I have to show you one other thing. So when they did this experiment, and they made that mouse called transgenic hell, they made that mouse in two different ways. And I've actually only told you about one of the two right now. The two different ways resulted in the protein being produced in two different ways. And so one of them resulted in this antigen being a transmembrane protein on cells. The other one resulted in this protein being secreted from cells. So here's my cell again, but here's my antigen. It's secreted. Notice it's kind of far away. Okay? So the version of the way part of this experiment that I have shown you thus far is the one where they put the antigen on the membrane. And in this case, demonstrated, they were able to demonstrate the receptor editing happened. Receptor editing. And let's just think a second about what's going on here. So this is my random cell from the mouse that has chicken protein on its surface. This is going to involve excellent artwork. Here's my B cell. You can see that this B cell is likely to have multiple B cell receptors triggered at the same time, right? Because you could have multiple B cell receptors binding to this multiple copies of the antigen. Because the antigen's kind of close together. It's all on the surface of the same cell. And so you could imagine this B cell gets a lot of signal. It gets a really strong signal, okay? In this situation, the antigen is secreted from the cell and is all kind of spread out around the mouse's body. And so as a result, we're probably only going to engage one receptor on that B cell surface. We're not going to engage a whole bunch of them. And so in this case, the cell is going to get a much weaker signal. So it only gets one arrow instead of a whole bunch of arrows worth of signal. So when they set up the experiment using the soluble HEL, turns out that they got a different result. They didn't get this same result. Instead, they got this result. So this was the data we were talking about before. Here is the data that they got now. So let's look at these data. So again, this is our mouse from the mouse store, the non-transgenic. This is the mouse that is the transgenic. This is our experimental mouse here, okay? So remember, if there was deletion, we would expect to see some non-B cells and no B cells. And we would expect non-B cells, and they definitely wouldn't bind to HEL. So this was deletion. Now, if we do receptor editing, we get some B cells, and those B cells don't bind to HEL. Right? No. B cells. We get some B cells. Those B cells don't bind to HEL. That's what we saw in the previous experiment. Ta-da! So what do they see in this experiment? Let's look at what's going on in these data. So it's this. Yeah. Are there B cells in these mice? Yes, there are B cells. There are cells that are positive for B220 in both cases. Do the B cells have heavy chains? Yes. B cell, yes. Heavy chain, yes. So our B cells have heavy chains. That's okay. That's what we saw in receptor editing. That's not crazy. What's going on here? Yeah, Sydney. Yeah, we got B cells that still bind hell. So this mouse still has B cells now that can still bind to this antigen. So in fact, in the case of this third option, we get this. Those B cells still bind hell. What? That's crazy. If we look at this mouse and we watch it run around the mouse cage, I should have told you this part before. If we look at the mouse and we watch it run around the mouse cage, it lives happily mouse ever after. It does not have mouse autoimmune disease. There is no mouse autoimmunity. So that was actually one part of this experiment. They made this. They saw the mice had no autoimmunity in any of the cases. So they knew tolerance had to go on. So this mouse is perfectly fine. It's running around, doing mouse things, being perfectly happy. But it has all sorts of B cells that are autoreactive, that bind to hell. And with further experiments, we found out about this third option. These flow cytometry plots tell us if the B cells are present or absent. They do not tell us if they actually do anything. And so you can imagine I could mark you present or absent for a class. But just because you're present doesn't mean you're functional. You know that feeling? So we know that the B cells are present with the flow cytometry, but in fact, those B cells stop being able to signal, and they become non-functional. We basically take that B cell and turn it off. So those B cells are all turned off through something called Anergy. And so basically, this cell is kept because it gets sort of a weak signal. It is kept, and it's allowed to leave the bone marrow off and completely unresponsive. We can talk a little bit more later about why you might keep that B cell and what the point of keeping that B cell is. But this third option, Anergy, is the third mechanism of peripheral tolerance in B cells. And we will finish up these slides and talk about the Ottinger paper on Wednesday. So I will see you guys then.